Awesome. All right, let me start. So, hi everyone. My name is Palash. I am a postdoctoral research associate at Princeton University, and today I'm going to talk about my research in computational biology. Obviously won't go into too many technical details about my research, but I do want this to serve as a primer for people who want to know about computational biology, what it is, why people are working in it, and what are the inner workings of doing research in computational biology. And when I was in high school, I did not know about such a field existing, and I wish I knew when I was in high school. So hopefully this will uh, be helpful for a lot of you. All right, so let's start with the most basic questions. Give me 1 second. All right, so what is computational biology? So I asked ChatGPT to answer this question for me, and it said that computational biology is an interdisciplinary field that applies computational techniques, algorithms, and models to analyze and interpret biological data. This is vague, but it will become more clear in the subsequent slides. And it merges principles from biology, computational science, computer science, mathematics, and statistics to gain insights into the complex biological processes and systems and structures all around us. And so the big takeaway from this slide should be that computational biology is very interdisciplinary. So if you are interested in multiple topics like biology and computer science, then computational biology might be the right fit for you. And also, this means that it's a place where people with different expertise and background can work together towards a common goal. So you really get to meet people with very diverse backgrounds, and they have very different way of thinking about the same problem, which makes this a very exciting field to work in. So why would you want to work in computational biology? So I'll go a bit personal here. So when I was trying to decide what I want to do for most of my life, I was trying to look at my academic interests. And at the time, I was interested in biology, of course, because one of some of the most complex and sophisticated machines out there are biological systems. The way our cells function, the way our bodies function was really fascinating to me as I was growing up. So biology was obviously a really fascinating topic for me and wanted to pursue that. But at the same time, I was really interested in mathematics. So at one point, I was thinking of going for med school because I was so interested in biology. But I realized that by doing that, I won't be able to do a lot of mathematics, which is also a really interesting topic for me. Uh, it's it's like a language that we use to describe the world around us, and uh, I was really in the at a fork with two paths. I thought with biology on one end and mathematics on the other. I didn't know if I could ever be able to do both. And after meandering for a very long time, because I didn't know if there was an option where I could actually do both, I ended up becoming a computational biologist. So if you are like me and if you like biology and mathematics both at the same time, uh, computational biology might be an interesting topic for you. So I want to uh, convince you of that in the subsequent slides. All right. Well, that was more of my personal motivations. Another reason to study computational biology is that it's a very fast moving and evolving field. There's so many things we don't know about the biology in the world. And computational biology is really um, um, emerging as one of the most promising tools to answer a lot of these questions. So Donald Knuth is a very famous professor in computer science at Stanford University. He is a winner of Turing Award, which is, if you don't know, it's like equivalent to Nobel Prize or something in mathematics. And he's called the father of analysis of algorithms. And this is a quote that he made a while back, and he said that he can't be as confident about computer science as he is about biology. So biology easily has 500 years of exciting problems to work on. It's at that level currently. So So that's another motivation to work in computational biology, would be at the cutting edge of discovery in biology. And another reason to work in computational biology is that it's really changing the way that we do biological discovery these days. So until the 20th century, both hypothesis generation and validation in medicine or biology was done using experimental techniques. So you would use microscopy or staining and other molecular experiments and try to understand the behavior of cells and tissues and so on, and try to generate hypotheses and then test them using these experiments. But since computational biology is becoming a big field and our computational resources are becoming more and more powerful, hypothesis generation I'm seeing more and more is starting to be it's outsourced to more computational side of things. So computational biology, we do hypothesis generation. We look at the data. We try to generate a hypothesis from that, what we think might be happening. And then the validation is something that's done on the experimental side more and more. And who knows, in the next few decades, you might see that even the validation side of things can be done on computers. So that's also a possibility. But for now, hypothesis generation is what computational biology is used for. And one of the reasons that computational biology has become so big in recent years is because the cost of sequencing has gone down drastically. So if you wanted to sequence the human genome, it would take you millions of dollars in the early 2000s. But now with technology becoming more and more efficient, you can sequence the human genome with for just like $1,000. And this reduction has been faster than Moore's law. And because of this, there's so much sequencing data out there. That's another reason that computational biology is an exciting field, because you have a lot of data to work with, and it's only growing by the day. It's becoming more and more, like governments are also starting to understand this, and you have programs at the government level to try to sequence their population, of course in an anonymous way, but try to get a large data bank of human genome sequences, and then you can then try to understand more about human biology, try to understand if there is some genetic factors that drive diseases and so on. All right, so let's start with this question about, I talked about how the cost of sequencing has gone down, but I never actually explained what does it mean to sequence a genome, right? So the human genome is very long. It's got 3 billion base pairs. When I say a base pair, it's almost like you can think of it as a letter. So if the human genome is a book, then each base pair is like a character. There are only 4 alphabets that make up the entire genome. That's A, C, T, and G. So you can think of the genome as an entire book, where pages of you have just these 4 letters in some order. And when we say that we are sequencing the genome, what we are really doing is that this human genome is cut up into very, very many tiny fragments. And these fragments are much, much smaller than the entire genome. So we are talking about fragments which are only 30 to 1,000 base pairs. So you can think of taking the novel, spreading all of its pages into small pieces, where each piece has around either 30 to 1,000 letters in. Okay, and then you have these machines that can sequence these small fragments for you. So they can read out the nucleotides in the small fragments for you. So for each of those small pieces of paper that you get from shredding the book, you can now read that it's okay, it's maybe CATP, CAG, and so on. Okay, there will be some noise when you read out these sequences also. But yeah, that's the kind of data that you're working with. So just imagine that you have, instead of having the entire novel, you have just these small pieces of paper. And that's the state of the art technology. And for a very long time, I don't think we'll get a technology that can actually sequence the entire genome from end to end. Okay, so the fundamental question in computational biology is then how do you even go from these small fragments to the entire human genome? And this is not a trivial problem at all. In fact, this should be one of the motivations that we do need uh, algorithms to make sense of this kind of data. And only once we get the entire genome can you even go into asking more detailed questions about the biology of the organism. Okay, so this is what it means to sequence a genome. Okay, and I want you to appreciate that the biological question of wanting to sequence the genome is very different from a computer science problem. So let's say that you are given these small reads and you want to assemble the genome. So that's called the genome assembly problem. That's a biological problem. As a computer scientist, you cannot just start working on this problem because it's not a well-formulated problem yet. So if you ever decide to come into computational biology, you would realize that biological problems are not the same as computer science problems. And the first task that you always do in this kind of research is to try to translate the biological problem in a mathematical framework. Okay, so let me give you one example of this translation. So I'm going to go from this genome assembly problem, which I hope you guys understand. So the entire human genome is very long. Current technologies do not let us actually read out the sequence from end to end for such a large genome. And so we have these small sections of the genome for which we have the readout information. And the task of genome assembly is, can you piece together these small sections and get back the entire genome that these small sections came from? These are called reads, by the way. So from the reads, can you assemble the entire genome from which the reads originated? That's the genome assembly problem. So the way that you can formulate this mathematically, and I'm not saying this is the only way, I'm saying this is one way of doing it. So when you do it, it should be a well-formulated mathematical problem. So you should have an input and you should have an output. And the input and output should be well-defined, okay? So you need certain abstractions to go from, some mathematical abstractions to go from these reads, which is a physical object, to something in math that you can play around with. So each read can be represented by a string, okay? So let's say that you have these strings over here, OWC, RTS, WCHAR, and FLO, okay? So these are the small reads that you have. There's a particular word from which these small substrings are coming, these reads are coming. You don't know what that word is at the moment because that's the problem. You want to assemble this word from these small strings. And the output would be, well, one way to formulate this problem would be, can you find me a string G that is the shortest common superstring of this set S. So what that means is, can you find me a string, a longer string, such that all these all these sequences that are given to me as input are substrings of that original string. So one solution for this particular problem would be flowcharts. So if you see all the different letters that all the different sequence of letters that were given to you as input are substrings of this word called flowcharts. So this is one way that you can formulate the problem of going from reads to the entire genome, assembling these reads together to get the entire genome. And I'm not saying that this is the only way to formulate this problem, but you would realize that there are certain assumptions that we are making. So first thing, we said that we want the shortest such common superstring. Why? Because of course, there is a very trivial way to generate a string. You just concatenate all of these small substrings in any order you like. And now you have a superstring where everything that was given to you as input is a substring of that superstring. And but that's not a meaningful solution because maybe I mean, you can, first of all, you can do it in many different ways, right? And also, you would expect that you should have some kind of objective function in your problem statement. So you want to, so in this case, for example, we are making an assumption that the shortest substring that has all of these, sorry, the shortest superstring that has all of these substrings is the most likely solution, or the most likely word from which these letters originated. Whether that is a reasonable assumption or not biologically is a separate thing. And applicability of an algorithm to solve this problem depends on whether that assumption that we are making about shortest being the most likely is true or not. Okay. So what, I mean, I hope you guys appreciate that this is a very difficult problem. Going from, I mean, what I mean by difficult is that it's not easy to start from a biological problem and then formulate a well-defined mathematical problem where the output will make biological sense. And there are many people who work on algorithms in computer science and stuff, but as a computational biologist, I think one thing that makes us different from the other computer scientists is that we need to bridge the gap between biology and computer science. So we need to be the people who would take a biological problem and formulate it well as a mathematical problem, and then also come up with nice algorithms to solve them. So one thing to always remember is that when you pose a computer science problem from a biological problem, it requires us to make certain simplifying assumptions. I never talked about the physics or the chemistry that goes into the machine that generates these reads and gives us the readouts, right? So obviously I ignored all of that. So I must be making some simplifying assumptions about how these reads were generated and what would be a reasonable solution to the problem. Even if you don't think you're making an assumption, you always are. So there's simplifying assumptions that you have to make. And the other thing is you need to have some kind of mathematical abstractions to go from a biological problem to a mathematical problem. Okay. So I'm not saying this is how the human genome was assembled, but you can think of it as the biggest yet jigsaw puzzle that has been solved by humankind up till now. This graphic here gives you the timeline of all the major developments that happened until 2021, when we got the first full genome completely sequenced. So from telomere to telomere, it's called. The telomeres are ends of our chromosomes. And what we have is now a complete sequence of a human genome. And this is actually a very recent development. It's only 2 years ago that we have the entire human genome sequence and assembled. So naturally, I'm sure some of you would ask, so are we done here? Like, is this it? Like, do we now have answer to everything? And the answer to that is no. Just because you now have the novel doesn't mean that you know the language in which the novel is written. So we have now, I would argue that this is the simpler part. Now we have assembled all the pieces of paper together to generate the entire novel from which we got all of this information. But now we have to read it and really understand it. And that requires us to understand language in which it is written, which is still an open problem. So in reality, we are just getting started here. And when I say that the human genome is sequenced and assembled, what does that even mean? There is billions of humans on planet. Whose genome have we sequenced? Whose genome have we assembled here? And the answer to that is there's no single human, I would think, that has whose genome is exactly the genome that we have assembled here. There are certain small changes in the human genome between individuals. So you must have noticed that we all look slightly different. And the reason for that is because we have some small changes between us in our genome. So you can imagine of the 3 billion base pairs in the human genome, around 10 to 100 million base pairs are different between any two humans. And so that's why they look different. But those human variations are actually very important. That those variations are what make you, right? There are different kind of these variations, and a lot of the different diseases actually happen because of these small variations, certain kind of variations. Okay, and so you might have blue eyes, and your friend might have black eyes. So reason because you have different colored eyes is because of these variations in the human gene. And I'm going to talk about, so now that we have the reference, we have the human genome assembled, what that means is now we have a reference to compare the sequencing information of any given individual to. So what that means is now you can take reads that you get from any sample. So let's say you take sample from my body, you run it through a sequencer, and you'll get a set of reads here. You'll get small fragments of my genome, and you can now read out the sequences of those small fragments. And now that you have the human genome assembled, the entire human genome assembled, you can now ask for each of these reads, where do these reads come from in the genome? And this is known as mapping the reads to the genome. Okay. And once you map them, you can also now see, let's say that this read is aligned to or mapped to this particular position in the genome. You can see if there are any differences between the assembled genome, which we call the reference genome, and the read that was aligned there. And those differences, those variations are what make each individual unique, and also might have the answer to some of the diseases that that person might be suffering. Okay, so I'll talk about three such variations, and that give rise to three different kind of diseases. So the first one is sickle cell anemia. I'm sure many of you might know about this, but what you might not know is there's a single nucleotide polymorphism. What that means is there's a variation in a single nucleotide in one of the genes called the beta globin gene that causes this disease. So what that means is that of the 3 billion base pairs in the human genome, there is a single base pair which, when it is different, when A is actually a T instead, when A turns into a T, that is what causes this disease. And I think that's fascinating that we were able to find that one difference in the genome that causes this disease. So even before the symptoms arise, if you have the sequencing information of any given person, you can actually tell whether they are going to have sickle cell anemia or not. The second example is color blindness. So I don't know how interactive we want to keep this, but just look at these images and see if you can tell the numbers inside each of these circle graphics. So certain fraction of people in the population will not be able to tell these numbers, and that's because of color blindness. So the numbers here are, there's 5 over here, there's 8 in the middle one, and there's also 8 on the right hand side here. Okay, and so up to and this happens because of a translocation in the X chromosome. And translocation is just when there's a jump in the genome. So oh, there's something in the chat. Just give me 1 second. I can't see the first one. All right, so you are one of the few who can't see some of these colors. I can't differentiate between these colors that well. We actually are asked to be quite mindful about these kind of things when we make results in our paper. So when we publish our paper, the figures that we put in our paper need to be such that they are, as much as possible, most people should be able to differentiate different colors in the figures so they can make sense of the figures. And so, yeah, so this happens because of translocation in the X chromosome. And typically 5% of people cannot spot the hidden colors in these cards. And usually these 5% are males. So it's interesting why this happens. I won't go into the biology of it, but it's very interesting. The next story that I have is actually, I find the most interesting story because there is something to say about the computational side of things in this story. Okay, there's a story behind this particular variation and this particular disease. So this disease is retinitis pigmentosa. So this is a photo of a normal left. On the left, you have a normal left eye. On the right, you have someone who has RP. You get these kind of pigmentation and these kind of spots on the eye. And it turns out that this particular disease happens because of a very, very tiny insertion, an insertion of only 353 base pairs. Okay, so just again, you have 3 billion base pair long genome, but just because of an insertion of 353 base pairs, you can get this disease called retinitis pigmentosa. And it happens in this MAC gene. MAC gene is much longer than 353 base pairs. I don't know the exact length of it in humans, but it's very long. Okay. And the interesting story about this is that when the researchers were trying to find these kind of variation in these genes, at first they were not able to find it. So because the software that they were using to align or map the reads, we just talked about this. So let me go back a few slides. So I told you, right, like we have the reference genome assembled 2 years ago. We have partially assembled it until 20 years ago, but now we have the full one. You can take reads that come from a particular individual and map them or align them, as we call it, align them to the reference genome. Okay, so this is an interesting story because it turns out the software that the researchers used to align the reads to the reference genome, that software made some mistakes. Okay, and so because of those mistakes, the MAC gene looked normal even for people who had the disease. And it was only because of pure accident that they were able to go back and see that, okay, some of the reads that were trimmed or discarded by the software actually had this information about 353 base pair insertion. And then later on, it was validated by orthogonal analysis. So if nothing else, this slide should really tell you how important the job of computational biologist is. Okay, the algorithms that we develop have real consequences. The accuracy of these algorithms are very important. For this particular case, it was fine because we were able to discover this, but it did prevent earlier discovery of this particular insertion that, which is the cause of this disease. Okay, so yeah, this is an important job. All right, and it's not a trivial job at all. These were smart people who wrote this software, but it's very difficult to make software that does everything perfectly all the time. So yeah, there's always going to be a moving target. Right. All right, so now I'll go more into what I like and what my work is more about. So this was just a primer. I won't be able to actually go into all aspects of computational biology because that's really big. It's like asking someone to give a primer of physics or something. So my favorite topic in biology is evolution. Okay? And when I talk to people about evolution, the first thing that comes to their mind is tree of life, that all the species on Earth are related to each other. And you can describe this relationship by a tree, which is why we call it the tree of life. Okay? And yes, this is a quintessential example of evolution in biology and a very important one. It changed our beliefs about humans and our relation to nature drastically in the last few centuries. But this is not the only example of evolution. And I would say that there's some other very interesting examples of evolution that people overlook. So in fact, I would argue that nothing in biology makes sense unless you look at it through the lens of evolution. And this is actually one of the most famous essays I've seen in biology. The title of the essay is "Nothing in Biology Makes Sense Except in the Light of Evolution." It's written by a very prominent geneticist and evolutionary biologist. I highly recommend you to check it out if you have a chance. This essay was written in 1973, and I think it's written for general public, so you should be able to get the gist of it. It's really interesting. So, apart from the tree of life, what other examples of evolution are there? Well, we just went through the COVID-19 pandemic in recent years, right? And I'm sure you guys heard about the virus evolving and new strains of virus becoming important, like the variant or the alpha variant, and then there was the Omicron, so on. That's one example of evolution. So the virus is evolving as it is being transmitted from one patient to another, one host to another during the outbreak. So that's one example of evolution. There's more. In fact, cancer is also an example of evolution. We call it somatic evolution. Okay. So tumors. Actually, I'll talk about cancer more in the next few slides. Even simpler example of evolution, although it's not really called evolution in the traditional sense, is normal development. So we all start from a single cell called the zygote, right? And this cell then divides until the present day, you have billions of cells that comprise your entire body. So all the cells in your body are actually related to each other. And this relationship, you can describe it by a single tree, because each cell divides into only two cells. You'll have a binary tree. By binary tree, I mean that each node only has two children. So these relationships about the parent and the daughter cells are being shown by these edges over here, these nodes over here, and these edges over here. So what this tree is telling you is that this is the zygote. It divided into two cells. These were the two cells, which again divided into two more cells each, and so on. Okay, so this is the cell lineage tree. And if you take all the cells in your body, you should be able to, I mean, in reality, there is some way of showing a relationship of all the cells in your body that can be represented by a tree that looks something like this. Okay. And even though you start from a single zygote, you get all these different cells that have very different function and they look different. So that means there is some change happening in these cells as they are dividing. They're what we call differentiating. They are specializing to have different functions. And one computational problem you can think of is, okay, let's say I get some sequencing information from all of these different cells. Can you then tell me how these cells are related to each other? And that's called lineage tracing. It's called tracing the lineage of the cells. Okay? I'll talk more about cancer in the next few slides. So cancer has a very, very similar process actually. Our current understanding of cancer is that cancer is an evolutionary process. Okay, so our cells in our body are dividing all the time. Okay, and whenever there is a cell that divides, your genome, the cell genome, has to be copied, right? And when that copy occurs, sometimes you can get errors. Cells sometimes make errors. There's a 3 billion base pair long genome, so a few places here and there you might get a mismatch. Okay. And those mismatches that happen during cell division in our body are known as somatic mutations. Now, most of these mutations don't hurt you, don't harm you. That's why you don't realize that these errors are happening as you go on in your normal day, right? But sometimes you can get an error, you can get a mutation that causes some problems. Your cell starts proliferating and starts behaving like a cancer cell. And this is how cancer starts. So here I'm trying to show a schematic where a single cell in this particular tissue, it acquired some of these mutations, which I'm depicting by a plus sign and a thunderbolt sign. And that mutation, these mutations changed something in the cell so that it started what is known as clonal expansion. So it starts proliferating more than it should. Okay, and doesn't. So our bodies regulate themselves. They don't let one cell just start dividing, because that would hinder the function of the tissue and cause death for other cells in the tissue and so on. So cancer starts like this. So you get a clonal expansion. And as those cells are dividing, the somatic mutations also keep accumulating. So as that happens, you get new somatic mutations that are acquired. Okay. And as this process occurs, you also get further clonal expansion. Sometimes the cancer cell can become more fit or start proliferating more than its parent. And so you get these clonal expansions. And so your tumor at present time then has subpopulation of cells. So here is the tumor, for example. You have this subpopulation of cells, which I'm showing with different colors, red, yellow, green, blue, and so on. that have different set of somatic mutations. Okay, so the cancer tumor is not a single thing. It's heterogeneous. It's got cells that are slightly different in their genomes. Okay, and there are different set of mutations in their genome, and this has huge clinical implications. Okay, so and the other thing is that the cancer evolution, it can be described by, again, a phylogenetic tree. This is how evolution happens. So you have your parent node that had certain somatic mutations where the cancer started, and then the daughter node that has some additional mutations, which were then passed on to its children, right? So, and then when you perform sequencing or when you take a biopsy from a patient in the hospital, you're basically taking a sample at present time that has cells from that have all these different kind of somatic mutations. Okay, and it's very important for biologists to know which of these mutations are present in the sample, and also what is the proportion of cells that have each of these mutations. Okay, and they want to know, because they want, and they want to know the evolutionary history of these different mutations. They want to know which mutation occurred first. Specifically, they would want to know which mutation started the cancer. So these blue mutations started the cancer. They want to know which are these mutations, where did they occur on the genome. They want to then know what are the different we call these subchronal, or which are the mutations that happen further down in the tree. Because you can imagine, if you try to treat the cancer in the patient, you try to remove some of these, kill some of these cells. You have to be careful that you kill cells that have all these different mutations, because unless you kill all of the cells, you will not cure the patient completely. And that's why you see relapse of cancer in patients, because some of these cells survive, and then because the treatment is not able to kill all the different kind of cancer cells, and so you get a new tumor in a few years. Okay, so this heterogeneity is very important to understand and to treat cancer. So when you sample this kind of thing, at present time, the way that sampling works is you get a mixture of these cancer cells. So you can take multiple samples from the same patient. It can be the same tumor or different tumors, and each of those samples you get you get this kind of reading that tells you what is the proportion of cells in that sample that had a particular mutation. So you get a matrix that looks like this, where you get fraction of cells with different mutations in different samples. Okay, and again, so now just think about it. This is such a difficult problem. Now you are just staring at these frequencies. You're staring at this fraction of cells that have certain mutations, and you're asked to find this phylogenetic tree, a tree on the where the edges are annotated by these mutations. Yeah, it's not an easy problem at all. And but if you make certain assumptions about how the cancer evolves, you can start to describe this in a mathematical framework. So the way we do that is we describe this phylogeny by a tree. Okay, a tree is just a graph where nodes represent the cancer cells and edges represent evolutionary relationship between these cancer cells. Okay, and then you can try to think about what are the relationship that these fractions or these frequencies should satisfy. So one relationship that I think all of you can very easily understand is that imagine that this is a particular cancer cell, and there's 80% of the samples, 80% of the cells in the sample have these set of mutations over here. Okay, and 30% have these set of mutations, 10% have these additional yellow mutations, and another 30% have these wild or purple mutations. So if this tree is really true, then you would imagine that the sum of these frequencies of the children should be less than or equal to the frequency of the parent. And here that is the case. So if you sum up these values here, it's 0.6 plus 0.1, that's 0.7, which is less than 0.8. But if this summed up to 0.85, then there's no way that these clones could have come after this particular clone, because this clone itself only has a frequency of 0.8. So this, we call it the nested structure. It's like Russian dolls, right? One doll inside another and so on. So I just wanted to give you a flavor of how math is used to try to describe the evolutionary uh, constraints on cancer clones. And this is the kind of thing that you would do if you enter computational biology. You'll try to think of different mathematical constraints and use that to go from these kind of information like frequencies of different clones in different samples and try to infer something about the biology, something about how cancer evolved. I think that's pretty cool. All right, so that's really the end of my talk. I want to end by saying that there's a lot more in computational biology. I only talked about a few things that I'm most familiar with and that I thought would give you a good introduction in this topic. But there's so much more. You can computational biology is used for drug discovery and design, personalized medicine, because treatment of like treatment with different drugs can affect people differently. And if you have more information about the genetic variations in a particular human, you can then, in a particular patient, you can then personalize their medicine protocol to take that into consideration and hopefully get better results. There's also other fields like microbiome and metagenomics where samples are taken from out in the environment. So, for example, you can imagine that you take samples from a lake. Okay, you get some water samples from lake and you sequence whatever genome is flying around or swimming around in that water, and try to tell me what are the different species of fish that live in that lake without ever having to actually do a surveillance of the entire lake, just by looking at a few um, flask of water, a few buckets of water. You should be, if uh, if you see the genome of that particular fish in the water, you know that that fish resides in that lake, right? So this is another kind of way in which computational biology is used. There's also a lot of work in protein folding and structure prediction of proteins, because in proteins, their function is directly related to the structure in which they fold. That's another thing I didn't talk about today, but also a very interesting topic in computational biology. So if you take computational biology in the first lecture of your course, you'll probably see some kind of uh, slides on all of these different problems. That's how they start to teach it. All right, that's all from my side. I'm happy to answer questions that you might have. Um, and yeah, thank you for listening.
I just want to say thank you for your lecture. I think a lot of people thought it was very interesting.
All right,
I really liked it. It was really good.
Yeah, the whole point of this was to so that you guys can become familiar with this field, because I didn't know about it at all in high school, and I wish I knew. I mean, I started in aerospace engineering, which is very different from what I'm doing now. And with time, I started to miss biology more and more, and so I made the switch. But if I knew about computational biology uh, earlier, then I could have made this. I could have started working on it in the beginning itself. So I think it's very important for one way that we can get more equity in students is if we get exposed to these different fields at the same time. So if you, yeah, the more people know about the different options out there, the better. I see some things in the chat. Let me see. Oh, there are no questions. Okay.
Yeah, I think there's a lot of room for like research and like new ideas that keep popping up with like the ideas of just using like computers in different fields. And I just like I don't I like the applications like in each field. It's like super cool. But yeah.
Yeah. Yeah. I also feel like it's so it feels very fulfilling when your work can be when you can say that your work is towards the betterment of human health and human condition. I think that can be a very fulfilling pursuit in life. And computational biology is becoming more and more popular. I know some of you might be applying for colleges this year or next year or something like that. And you should definitely look up if you're interested in this kind of work, even if you just want it as an option later on. You don't maybe know for sure whether you want to do it immediately. You still have time. You're just still students like you. you have your whole undergrad to figure out what you want to do. But computational biology is becoming a field that almost all computer science departments now have some people working in computational biology. Some universities even have their own departments dedicated for computational biology. And another thing they call it is biostats. So biostatistics and computational biology. Those are the keywords you want to look out for when you're applying for colleges, if you're interested in this. Are there any new up and coming technologies for computational biology? That's a great question. Yes, there are. In fact, one reason that I like computational biology is that our work is always driven by technological developments in that amazing people in experimental side of things do. So let me give you an example. So in the last 5 years, you will see there's new technologies coming out that are known as spatial molecular profiling technologies. Okay? So in these technologies, what you're able to do is you're able to measure the DNA, RNA, or even proteins of cells in a tissue, but you're able to do it in a way that you keep the spatial context of these cells intact. So what that means is that if you take a particular slice of a tissue, like let's say you take a piece of liver from a patient, you can now sequence it in a way that you can tell, okay, this cell over here has this particular protein, this cell has this much abundance of this protein. You can do that while keeping the spatial locations intact. And what that, so with at a very high resolution, you're able to get these measurements. And why that is a good thing is because cells, the spatial organization of cells is very important for the function of the organ. So because cells communicate, like neighboring cells will talk to each other and exchange resources and so on. So these are the new technologies. This is just, it's must sound, I mean, to me, it really sounds like magic that you're able to do this. Like experimentalists are actually able to measure these things. And that's the exciting part. Like you have all these awesome people creating these new experiments, and you get this new kind of data. Now you have new algorithms to develop and play around with new kind of data all the time. I don't know of any other field where you get to work on these exciting different modalities of data all the time. What classes or resources did you reference or take when you got started in your career? Oh, there's one more question. What type of technology and different exists for biologists to use to deduce evolutionary tree of a single cancer symptom? Okay, so the first question about what type of technology and different experiments exist for biologists to use to deduce evolutionary tree of a single cancer symptom. So the answer to that is that you have the most recent kind of technology that people have started to use now is known as single cell sequencing. So in single cell sequencing, what you do is you take single cells from the tumor, you isolate single cells, okay, and you can independently sequence the genome of each of these cells. Okay, so now you have sequencing information, you have sequencing reads from, you can get, I think, even 1 million cells these days, but let's say you get a few thousand cells. Okay, so for each of those cells, you know the sequencing reads. And now you can try to ask a very simple question. All of these cells have come from the same tumor, right? And all the whole tumor must have started when a single cell acquired the somatic mutations. So the first question you can ask is, how are these cells related to each other? Can you tell me the entire evolutionary tree of all the cells in this particular tumor? So yeah, that's the kind of technology. If you want to Google, it would be single cell sequencing. Just if you Google that, you'll be able to see what kind of technologies are out there. What classes and/or resources did you reference or take to get started in your career? Okay, this is a good question. So if you are interested in computational biology, actually, I would like to start by saying that computational biology is very, very big. Okay, you would want to have a good background in mathematics and computer science to be in computational biology. And the reason I did not mention biology itself is because as you work in this field for long enough, you start to learn enough biology to formulate problems, but you'll never become a full biologist. So for example, I am a computational biologist, but I think I'm more computational than biologist. There are some people who are in computational biology where they come from biology background, and some people who are more computer scientists who like biology. Okay, so like if you want to do the kind of work that I'm doing, it will be more that you need to have a good background in computer science and mathematics. And the kind of math that I use more now is discrete mathematics. So discrete structures. The reason for that is because a lot of things in biology also have discrete structures. So discrete mathematical abstractions are very important. So what I mean by that is that the genome has only 4 alphabets, A, C, T, G. So 4, that's a discrete number, right? Proteins, you have 20 amino acids or so. So you have all these different amino acids, there's only 20, right? What else? So
sorry to interrupt. I think it might be my end, but I'm not able to hear you.
hello?
Yeah, there you go. Hello.
Hello?
Yeah, are you able to hear me?
Yeah, yes, we can hear you. At least I can.
Okay. Okay, great. Sorry about that. I don't know where I got cut off, but okay. So there are 2 different camps of computational biology. One where you would use more discrete mathematics. So that's graph theory and combinatorial optimization. You can look up those kind of things. On the left-hand side, there's also systems biology, where you want to use differential equations. So more continuum side of mathematics, okay? So multivariate calculus, differential equations, Riemannian geometry, that's one side. On the right-hand side, also the discrete mathematics, you have combinatorial optimization, graph theory, and what else? Yeah, computational geometry is also used. So that is to say that whatever background you have in mathematics, you should be able to do some problems in computation. Is set theory involved? Absolutely, yes. Set theory is also involved. Although, I mean, my work does not use set theory so much, but yes, set theory is also involved a lot in computational biology. Again, computational biology is a very big field, right? So there are certain things in which continuous mathematics can be used, continuum mathematics can be used. So if you think about gene expression, okay, how much gene is being expressed in a particular cell, that's more in a continuum, right? But if you want to talk about the genome assembly problem that we talked about, where I talk about strings, that's more discrete because you only have A, C, T, G, so you only have 4 alphabets. I think honestly, if you want to know about different resources you need, whenever you take these courses, you'll always see some kind of prerequisites required. So they'll tell you what kind of courses you require to, like what kind of background you need to have to get the most out of that course. So you'll be able to see that if you decide to go in this direction in your university. All right, thank you for the question. I'm still not sure if you guys can hear me, so just
Yeah. We can hear
okay,
you.
good.
Yeah.
Awesome. All right. Okay. I don't know what happened. My sharing also stopped suddenly. It got paused and stopped. So I don't know. That's why I was not sure if it was. Great. If you guys ever, any of you ever have any specific questions about Princeton or computational biology in Princeton, you can ask me or contact me later. It doesn't have to be now. Because I know some of you will be applying soon. And what else? Yeah, that's all I have in mind. And you have plenty of time to decide this. Computational biology is one of those things that you can enter late also, because so many different, there is some place for everybody with different skill sets because it's so interdisciplinary. So I always feel like, okay, even if you are a physicist, you can do something in computation. Even if you are a biologist, you can do something. So it's there's always room for someone in this field.
Okay, thank you so much. Your talk was great. I think everyone learned a lot, and it was really a great introduction to computational biology.
All right, thank you.